So hello everyone, today we are going to discuss about the heat shock proteins. So before going to the heat, exactly what is the heat shock proteins? Scientists were carrying out experiment before 1960. They observed that when the plants are growing under high temperature conditions, these plant doesn't show that much of alteration in or these plants doesn't show alteration in the gene expression. So they realize that there is some mechanism which is helping these plants under the stress condition. So they observe in the Drosophila first in 1960s, they observed that there are some proteins which help native proteins or the cytosolic proteins not to get altered, not to get altered. So they called it as the heat shock proteins. They call this as heat shock proteins. Then later, these heat shock proteins were also observed in E. coli, Saccharomyces cerevisiae, as well as in the humans. These heat shock proteins are having they are having conserved sequences in their own, like they are having conserved sequence either in plants, they are having conserved sequence in case of humans as well. So later on, when they were doing research on these small proteins, they observed that whenever the plants are allowed to grow above their optimum temperature, so that time these proteins were expressed. So they have observed if we will grow plant if we will grow plant exceeding the temperature 32 to 33 degrees centigrade, that time these heat shock proteins are synthesized. Means these heat shock protein are developed when thermotolerance, or these heat shock proteins are having thermotolerance in nature. So these heat shock protein has been observed in different variety of plant species like soybean, maize, rice, wheat. So these heat shock proteins are designated as per, or they are classified as per their molecular weight. Molecular weight is in carried is the unit of the molecular weight is kilodaltons. So whenever we are seeing that the heat shock protein 10 or 110, we can say the molecular weight of this protein which we are talking is the 110 kilodaltons. Like we are having different heat shock proteins, heat shock protein 110, heat shock protein 19, heat shock protein 70, 60, and so on. And plus we are having some small molecular weight heat shock proteins. Their molecular weight varies from 15 to 30 kilodaltons. I will give you one example. Like you have seen in electron transport chain, we are having ubiquitin molecule. This ubiquitin molecule is not the part of the any complex there. We are having four complexes in case of mitochondria, electron transport chain. So in case we are having small molecule ubiquitin which takes electron from complex 1 to complex 3, or from complex 1 to complex 4, these proteins have or these proteins come under the heat shock proteins. So you have all realized that what is ubiquitin protein. So if we will talk about this now, this was the earlier concept of the heat shock proteins. Later on it was observed that these heat shock proteins are not only involved in the these heat shock proteins are not involved in the during the high temperature only, but they give resistance to the plants under different environmental conditions. Means we initially these were observed during the heat shock, but later they observed that these proteins are also expressed under stress condition. It may be the chilling temperature, it may be the salinity. Means under all stress conditions these heat shock proteins are expressed in plant cells in higher amount. So if we will talk about now in plants we are having small heat shock protein as a major class of heat shock proteins. Again, I will repeat. In case of plants, we are having small heat shock proteins as a major class of heat shock proteins. As I said, they are highly conserved. These heat shock proteins are mostly composed of 90 amino acids, and at their C terminal they are having long alpha crystalline domains. So if a layman will ask what are heat shock proteins, you can say these are conserved proteins, highly conserved. They are having 90 amino acids in length, and at their C terminal they are having alpha crystalline domains. If we will talk about the monomeric molecular weight of the heat shock protein, that comes in the range of 15 to 40 kilodaltons. But heat, as name indicates, it's a protein, so they are largely composed of oligomeric assemblies, means there is more than one heat shock protein involved in, or there is more than one monomeric unit present. Till now, we have observed 6 nuclear families in plants for these heat shock proteins. Nuclear families means these heat shock proteins are repeatedly present in their genome. As we know, in case of ribosomal RNA, nucleolus, there also we are having gene families for ribosomal RNA. means इसका मतलब क्या हुआ कि जो ये हीट शॉक प्रोटीन्स के जो जीन्स है they are arranged in repeats. These repeats are continuous, so in all together we call these as families, gene families. So it has been estimated that for heat shock protein in case of plants we are having 6 nuclear gene families. These जो ये 6 nuclear families है, they are having sequence similarity. They are having sequence similarity with each other, and it has been observed among the 6 nuclear gene families we are having 2 gene families which are localized to the cytosol only. 6 में से हमारी जो जीन फैमिलीज है उसमें से जो 2 है वो किसके लिए है? That is for the organelle, that is for the cytosol localization. मीन जो हमारी 6 जीन फैमिलीज है हीट शॉक के लिए, आउट ऑफ 6 वी आर हैविंग 2 heat shock proteins which are only meant for the cytosol only. These gene families were designated as C1 and C2. These two cytosolic localization sequences are identified as C1 and C2. If in the future, during this lecture, we will come across with the C1 or C2, it's obvious these are the heat shock proteins which are for cytosol. So what they do, what is the main function? If we will talk about the functioning of the heat shock proteins, what they do under stress conditions, the proteins which are in the cytoplasm or which are going to be transferred to any organelle from the cytosol, these under high temperature there is the probability that these protein will get aggregated or they will get folded up in the cytosol only. If these proteins which were designated from cytosol to the different organelle will get in their midway, they will get folded up, then the whole mechanism for the metabolic process will get hampered. So what they do, these heat shock protein will come to get attached with the cytosolic protein, so that under stress condition as well, these proteins should not get folded up. As if you remember, we have discussed about the heat shock protein 70, HSP 70, when the cytosolic protein was transferred from cytosol to mitochondria. We have discussed this thing in our previous semester, right? Then within the inner membrane of the mitochondria there was HSP 60, which were allowing it to fold in a proper way. This example we have studied earlier, but today we are discussing only about the heat shock proteins in case of plants. So it has been seen that these heat shock proteins have role in diverse stress conditions. It's not only the in under heat shock. No, they are having diverse. They are involved in the diverse stress conditions. Like okay, if there is a temperature decrease in the temperature, like cold is there, dehydration, salinity, or even the oxidative stress due to the metabolic processes. There are presence of free radicals, so they that the those free radicals increase the oxidative stress in the plants. So there comes the role of the heat shock proteins. So if we will say about the heat shock proteins, it has been now observed that the heat shock proteins are involved in multi purposes. They are having different role in different developmental processes. As we can see, overall the heat shock proteins are overall involved in the developmental process of the plant. They are involved when the pollens are formed. We can simply say pollen development. They are involved in the seed development or storage organelle in case of seed. Or we can say the role of small histone protein in maintaining seed viability during aging. As we keep seeds for longer time, there is aging process occurs. Then if there is not the presence of heat shock protein, then those seeds will not remain viable. And under stress condition, these heat shock proteins allow seeds to germinate and allows these seeds to germinate in a better condition. So if we will talk about the general if we will generally talk about the role of heat shock proteins in the developmental process in the plant, they are involved not only in the increased temperature, but they also allow plant to repair under the stress conditions. Like if we will see here, the heat shock proteins in case of seed maturation, when there is seeds are getting mature in the plants, that time there is higher expression of heat shock proteins. So this heat shock proteins for seed maturation has been observed in different varieties, like in Pisum sativum, Triticum aestivum, or Glycine max. Or now if we will shift to the heat in role of small heat shock proteins in the pollen development, we will be having best example is the Zea mays or Nicotina tabacum. Or if we will talk about the role of small heat shock proteins in the storage organelle, we are having different examples there, like Tulipa is there, Corocus is there. These are different flowering variety. These are the different flowering variety where small heat shock protein are involved in storage organs. Or if we will talk about the heat shock protein in embryo development, it has been seen in the Arabidopsis thaliana, Pisum sativum. So when we are seeing the role of small heat shock proteins in seed germination, it has been observed in Pisum sativum or Arabidopsis thaliana. In general, we can say that we are having many examples where these heat shock proteins are overall involved. Now, the heat shock protein in the pollen development. So class second small heat shock protein. Again, class second. It indicates what? These are the proteins which are localized for the cytosol. So for pollen development, it has observed that the class second small heat shock protein are expressed more during the pollen development in case of lily and in case of peas. In case of lily, not peas, in case of maize. Lily and maize are the examples where it has seen that the small heat shock proteins are there. They belong to the class second. So while pollens are developed during their mitotic stage, these HSP are in higher number. So the small heat shock protein which are in higher number in pollen development are heat shock protein 18. So if I will ask you in the exams which heat shock protein is involved in the pollen development. It simply indicates that heat shock protein 18, and this heat shock protein is observed in lily as well as in the maize. And this was about the plants. If we will talk about this heat shock protein, HSP18 has been also seen that they are higher expressed in yeast as well. Means HSP18 is also expressed in yeast. Best example is among the yeast is the Saccharomyces cerevisiae. It has been observed during the late prophase of meiosis. लेट प्रोफेज ऑफ मियोसिस के टाइम पे HSP18 को देखा गया है. इवन we have seen this HSP18 in other plants as well, as it is also seen in the Drosophila. Plants में देखा गया है. In short, we can say HSP18s are involved in different plants, yeast and Drosophila, during the having the role in the mitotic cells. Having the role in the mitotic cells either in anthers or in the pollens में, in short. This was about the role of heat shock protein in the pollen development. So we can say class second cytosolic proteins which have been observed in lily as well as in the maize. The important heat shock protein which were observed during the pollen development is HS18, and the higher expression of HS18 were also observed in the yeast during the late prophase of meiosis. Now, if we will talk another example, the role of small heat shock protein in the seed development. The first report for the seed development has been come from the Pisum sativum in the year 1994 and 95. What? The role of HSP in the seed development were first reported from the Pisum sativum by the two scientists D. Rocher and Verling in 1994 and 95, where they observed that we are having higher transcript of HSP in the cotyledon of pea seed. जब seed develop हो रहा होता है उस टाइम पे what they have seen that the higher transcript for HSP has been observed during the cotyledon of pea seeds. जो ही ये जो start and this HSP get declined when the seed starts to germinate. Means before germination, when the seed has to germinate, before the seed germination there is a higher amount of HSP in the cell. जो ही इस seed ने क्या किया, imbibe पानी लेना शुरू किया, imbibition किया, फिर उसके बाद क्या होगी, there are the processes which will allow this seed to get germinated. As we know, in the seed we are having three important components. One is the plumule, another is the radicle, and the third component is the endosperm. This endosperm will act as a nutritional point for the growing of the plumule and the radicle. जो ही इस seed को हमने sow किया under artificial condition, that time we have seen that there is a higher amount of heat shock proteins in these cotyledons of pea seeds, and it declines. ये जल्दी खत्म हो जाता है as germination starts. So we are having cytoplasmic class of small heat shock proteins. We are having cytoplasmic class of heat shock protein which get accumulated during the high temperatures. If we are allowing seeds to grow under high temperature condition, this experiment has been carried out in P and alpha. In case of P and alpha, when their seeds were allowed to grow under high temperature conditions, there is the higher accumulation level of cytoplasmic classes of small heat shock protein. It means अगर ये seed था one is अगर हम लोग ये seed को ले रहे हैं, seed from P and alpha, if we allow it to develop under high temperature conditions, we have observed that there is the higher accumulation of cytoplasmic class of small heat shock proteins. So what it indicates? It indicates that when the seed is under the heat shock or when there is a higher temperature, that time cytoplasmic class of small heat shock proteins are expressed. So so like here we have seen, we are having proteins. If there will be no heat shock protein, then there will be the aggregation of the proteins, so that will hamper our metabolic processes. But when there is the heat is temperature is given, it means plant is under stress condition. Due to this stress condition, there is the higher expression of small heat shock proteins. These heat shock proteins get mixed up with the cytosolic or the native proteins of the plant and doesn't allow to get aggregated with each other. This is how the heat shock protein works together. Now we are having the role of small heat shock protein in the seed germination. this heat shock protein has been observed in sunflower. This heat shock protein has been observed in sunflower as well as in peas. In sunflower, they have observed two different types of heat shock proteins. First is the heat shock protein 17.6 and the another protein heat shock protein 17.9. जो ही इस प्लांट को, इस सीड को क्या करना है, जर्मिनेट करना है, उस टाइम पे ये होते हैं. जो ही इसने जर्मिनेशन स्टार्ट की, इसकी जो प्रोटीन इसकी जो ट्रांसक्रिप्शन है वो स्टॉप हो जाती है. बट जो ही बिफोर द जर्मिनेशन they have raised their number in the seed up to so much that their proteins remain there for 3 days after the germination. Like वे simply मैं simple sense में बोलूँगी, जब seed को क्या करना है, germinate करना है, before germination there is the higher accumulation of heat shock proteins. जो ही germination start हुई, उस टाइम पे क्या होता है, plant heat shock proteins नहीं बनाता है. लेकिन ऐसा देखा गया है इन केस ऑफ सनफ्लावर में कि ट्रांसक्रिप्शन तो बंद हो रही है, लेकिन जो प्रोटीन्स है सनफ्लावर में वो 3 दिन तक वहाँ पे रहते हैं, क्योंकि बिफोर जर्मिनेशन उनकी इतनी ज़्यादा फॉर्मेशन हुई होती है. एटलीस्ट इट टेक 3 days टू ओवरकम दीज प्रोटीन्स. 3 दिन तक वो प्रोटीन्स वहाँ पे रहते हैं. तो सिमिलरली इसका जो सीड जर्मिनेशन का रोल देखा गया है इन केस ऑफ peas as well. They are also they exist in the cotyledons and they remain there for these proteins remain there in the cotyledons after the imbibition of water as well, and they get declined before the germination of the seeds start. Means imbibition के टाइम पे हायर एक्यूमुलेशन रहता है, जो ही सीड जर्मिनेट होता है उस टाइम पे वो ख़त्म हो जाता है. So now we will see the role of heat shock proteins in the storage organs. As we know, in plants we are having many storage organs, like bulbs are there, or we are having tubers. So we are having 3 different example I have given you: alimony, coccus and tulipa, and the potatoes are there. What they have seen, when there is the accumulation, हमेशा सीड में क्या होता है, सीड इज़ ऑलवेज हैविंग accumulation ऑफ स्टोरेज प्रोटीन्स. ऐसा देखा गया कि अगर सीड में स्टोरेज प्रोटीन है, उस टाइम पे हीट शॉक प्रोटीन्स भी प्रेजेंट होते हैं. ठीक, मीन्स इट्स वाइस वर्सा. when there is the accumulation of storage protein in the seed, that time small HSP proteins are there. Now they have carried out an experiment, क्योंकि हर एक एक्सपेरिमेंट का हमें प्रूफ चाहिए होता है. They have carried out an experiment on transgenic tobacco. उन्होंने उसके एम्ब्रियो में देखा है कि अगर उसका एम्ब्रियो लैक करता है प्रोटीन बॉडी रखने में, या there is no accumulation of storage protein in the embryo of the transgenic tobacco, that time that embryo is not having accumulation of small heat shock proteins. Means इसको simply अगर बोला जाएगा कि जब प्रोटीन accumulation हो रही है कहाँ पे, सीड्स में, उस टाइम पे हीट शॉक प्रोटीन्स भी accumulate हो रहे हैं. ठीक, अगर इन्होंने दे हैव कैरीड एक्सपेरिमेंट ऑन ट्रांसजेनिक टैबैको, टैबैको में इन्होंने क्या देखा कि टैबैको में उन्होंने ऐसा मैकेनिज्म रखा कि जिसमें एक्यूमुलेशन, प्रोटीन एक्यूमुलेशन नहीं होगी, वहाँ पे स्टोरेज ऑफ प्रोटीन नहीं होगी। वहाँ पे फिर देखा उन्होंने कि अगर प्रोटीन स्टोरेज नहीं हो रही है तो वहाँ पे स्मॉल हीट शॉक प्रोटीन्स भी नहीं है। मीन्स इफ proteins are getting accumulated, then sidewise we are having accumulation of heat shock protein as well. Even if we will talk about the simple, these heat shock protein जब ये express करते हैं ना, they express up to 20,000 copies per cell. 20,000 copies of mRNA transcript means 20,000 proteins बनाते हैं within in a cell. To get attached these proteins to the target protein of the cytoplasm, they need some मतलब बाइंडिंग अटैचमेंट। मीन्स एक प्रोटीन जो नॉर्मल साइटोसोलिक प्रोटीन है जिसको डर है कि वो फोल्ड हो जाएगा, वहाँ पे क्या आएगा, हीट शॉक प्रोटीन आके एक्टिवेट करेगा। वहाँ पे हीट शॉक एलिमेंट्स आते हैं, जो हीट शॉक एलिमेंट्स बाइंडिंग साइड बनाते हैं हीट शॉक प्रोटीन्स के लिए। So now we will see the role of small heat shock protein in maintaining seed viability during aging. It has been seen that these heat shock proteins are having ability to initiate the seed germination or to maintain the seed viability under stress condition as well. What they have observed, they have taken one transgenic plant in which they have observed the overexpression of small heat shock protein under different stress conditions. What the scientists have do, they have taken a transgenic plant in which they have given different stress conditions to the plant, by in which they have observed that there is the overexpression of हीट शॉक प्रोटीन्स। जब हम लोग ट्रांसजेनिक प्लांट को डिफरेंट स्ट्रेस कंडीशंस देते हैं उस टाइम पे क्या होता है, उस टाइम पे देखा गया है कि वहाँ पे ओवर एक्सप्रेशन ऑफ स्मॉल हीट शॉक प्रोटीन्स होते हैं। दे हैव गिवन डिफरेंट कंडीशंस लाइक क्या उन्होंने देखे, ऑस्मोटिक स्ट्रेस टॉलरेंस देखी, उन्होंने चिलिंग टॉलरेंस देखी, थर्मो टॉलरेंस देखी। इवन उसकी वजह से क्या हुआ, सीड की जो लॉन्गिटिविटी है वो इंक्रीज हो गई। ठीक, it means under these 4 different circumstances heat shock proteins were expressed in a very larger amount, but इन्होंने देखा है अगर ये बाकी कंडीशंस में इसकी जो है मतलब हायर एक्सप्रेशन हो रहा है, लेकिन यही प्लांट जो है जब हम इसको हीट शॉक देते हैं या थर्मल शॉक देते हैं उस टाइम पे इसकी टॉलरेंस बहुत कम हो जाती है, और ये जो प्लांट्स है ये ससेप्टेबल टू पैथोजन है। अगेन आई विल रिपीट, दे हैव कैरीड आउट अ एक्सपेरिमेंट जहाँ पे इन्होंने प्लांट को एक्सेप्ट हीट शॉक सारे एनवायरमेंटल स्ट्रेस कंडीशंस दिए, लाइक थर्मो टॉलरेंस देखा इन्होंने ऑस्मोटिक स्ट्रेस टॉलरेंस देखा, चिलिंग टॉलरेंस देखा और सीड लॉन्गिटिविटी देखी। इन्होंने देखा कि ऑल द 3 अंडर ऑल द 4 कंडीशंस देयर इज़ द ओवर एक्सप्रेशन ऑफ द स्मॉल हीट शॉक प्रोटीन्स। जब प्लांट एक डायरेक्शन में दे रहा है ना ओवर एक्सप्रेशन, जो इसका मेन फंक्शन है हीट शॉक थर्मल शॉक, ये उस टाइम पे इसको अपनी कम कैपेसिटी दिखा रहा है, या तो ये कम्प्लीटली जाएगा हीट शॉक की तरफ या तो ये फिर प्लांट्स को दूसरे स्ट्रेस कंडीशन से बचाता है। और इन्होंने देखा गया है कि जब ये प्लांट दूसरे स्ट्रेस कंडीशन में जा रहा है इसमें थर्मल शॉक की कैपेसिटी नहीं होती है, और प्लस इसमें दीज प्लांट्स आर ससेप्टेबल टू द पैथोजेंस। So the role of small heat shock protein in better germinability to seeds under stress conditions. Simply, जब what they have done in this मतलब they have seen that when the seeds were allowed to germinate under stress conditions, again they have observed that there is the overexpression of certain heat shock proteins. This experiment were carried out in rice, where they have observed when this rice plant were taken under the stress condition. These under stress condition as well, these seeds were able to germinate properly. How? They have observed there two different types of heat shock proteins. They have called these heat shock protein as OSHP17 and another is OSHP23.7. As we know, these HP17 will be as per their molecular weight. This is how plants are developing, or plants have developed different environmental conditions due to the presence of the heat shock proteins in them.